Hi, thank you for that kind introduction. So my talk today is called Exploring the Unexplored, Radiometals for Theranostics. So as we all are aware in this audience at this point, radiopharmaceutical therapy is rapidly emerging as a potent, effective, and well-tolerated approach to treat patients with advanced metastatic cancers. The image shown here is one from British Columbia Cancer Agency's clinical trial using a beta-emitting lutetium agent to treat patients with advanced metastatic castration-resistant prostate cancer. So mostly all of the radiopharmaceutical therapies under development today require a radioactive metal ion as the active ingredient. The principles of radiopharmaceutical therapy are simple, relying on radioactive drugs that bind tightly to, that bind tightly to proteins that are selectively overexpressed on the surface of cancer cells. Using a radioactive metal ion, we can incorporate these into the drug via the bifunctional chelator strategy shown here, which requires a molecular cage called a chelator to bind that metal ion in a stable complex, ensuring the delivery to the site in vivo. This is very modular, so we can swap out the radiometal with one that emits isotopes for imaging, for PET or SPECT, or another isotope that emits therapeutic radiation via emission of beta, alpha particles, or minor Auger electrons. This is amenable to the theranostic approach, where we can treat what we see and see what we treat. Treat what we see and treat what we see. So in terms of therapeutic isotopes and the emissions associated with them, we ensure that the isotope has the particles that can be effective at killing cells close to where it's emitted. And most of the most advanced clinical trials with lutetium, for example, are beta, which is a beta emitter. However, alpha particles as well as minor Auger electrons have a lot more cytotoxic punch, which can deliver an even more effective dose to your cancer cell, even closer to the site of decay, ensuring we have a more effective therapy and less resistance to develop resistance in the future. Another advantage of the radiometals in theranostics is that all of the long synthetic preparation to make your chelator, conjugate that to your biomolecule, this all happens ahead of time. And the last step is adding your radioisotope, saving many half-lives of radioactivity. So this reaction, the radiolabeling step at the end, can be optimized so that it's quantitative, done under mild conditions, such that at the end we don't need any final purification of the tracer, and this rapidly accelerates the adoption of these tracers in clinical radiopharmacies. This is what we call kit-type labeling and what I call shake-and-shoot chemistry. So in terms of radiometals, we really have unmatched diversity in terms of physical half-lives and decay emissions, meaning we can exploit all the modalities possible with nuclear medicine and therapy. Even further still, several elements have multiple isotopes that can be used for imaging or therapy, meaning we can match a diagnostic isotope of a chemically identical or chemically similar element with one that emits therapeutic radiation. This is important because we can use the same chelator and thus the same molecular scaffold for both imaging and therapy. This ensures our diagnostic imaging will accurately reflect the therapeutic response. So some examples of other theranostic pairs are shown on this slide. And we really work across the periodic table to exploit this nuclear chocolate box. So our lab, as well as 18 other PIs across Canada, are currently working on a funded program through the New Frontiers in Research Fund called Rare Isotopes to Transform Cancer Therapy, with the ultimate goal to transform cancer radiopharmaceutical therapies and theranostics in Canada. Some metrics of the program are shown here. Our team integrates all key aspects in order to bring therapies from beamline to bedside. So we are an interdisciplinary team from diverse backgrounds across British Columbia, Alberta, Ontario, and Quebec. And we have all the integrated expertise, including basic science, clinical translation, as well as social and health impacts. Our approach is devised over 5 integrated and cohesive aims. In aim 1, we produce a library of isotopes which we believe have the strongest potential for clinical impact. In aim 2, we use our world-leading research in radiometal chelator design to optimize the chelator to bind all the isotopes produced in aim 1. In aim 3, we develop novel radioligands that have binding properties to various cancer cell receptors. We're working on targets targeting breast cancer, melanoma, FAP, colorectal cancer, to name a few. In aim 4, we initiate investigator-led clinical trials. Our knowledge mobilization and patient engagement committee has given the green light to commence 2 early phase trials targeting FAP and breast cancer at the BC Cancer Agency as well as University of Toronto. And aim 5 is integrated within this translational pathway and uses regulatory science and social and health economic impact analyses to understand the true impact of radiopharmaceuticals in Canada. So these studies really help direct where our project is going in terms of which isotope do we prioritize, how do we design our clinical trials, as well as ensuring that these products have adoption within the clinical setting and are accepted by patients. So in terms of our isotope production capabilities, we leverage unique particle accelerator infrastructure across Canada. For example, using Triumph's 500 MeV cyclotron, we can produce an array of very potent alpha emitters such as thorium, actinium, bismuth, and lead in large quantities. And by leveraging all the small titanium cyclotrons across our consortium, we can produce a variety of other medically relevant diagnostic, therapeutic, or theranostic isotopes. Many of the metals that we work on remain largely unexplored, so we require a lot of innovation in terms of designing first-of-class systems that are able to bind these isotopes and effectively deliver them to cancer sites. And 2 examples are shown on this slide for isotopes of mercury-197 and the lead-203-212 theranostic pair. So in summary, we believe by integrating expertise across basic science, clinical translation, and social and health economic impact analyses, we'll ensure that we will be successful in translating theranostics in Canada in the next 6 years, as well as training the next generation of HQP that will be well suited to fuel the growing industry of Canadian startups and companies. Thank you.